It is a distinct pleasure to welcome the wrap-up speaker for session number 2 on technological pathways for medical nanobots. The wrap-up speaker comes to us from the institute founded by the first conceptualizer of the entire field of nanomedicine, Eric Drexler. And Eric Drexler published his pioneering work in the late 80s and early 90s and formed the Foresight Institute. And our speaker, Tihamer Toth-Fejel, comes from the Foresight Institute. He has had a really illustrious career of doing a lot of great and exciting work in nanotechnology prior to that. But I'm especially excited that Tihamer is now working with the Foresight Institute and is sharing the research that he's been doing there with us this morning. Please join me in a warm welcome for Dr. Toth-Fejel.
First of all, thank you, Martine and Baruch, for this great conference. What I'm going to be talking about is actually, Eric Drexler didn't talk about this, but one of his first people that started popularizing and writing about nanotechnology afterwards is Robert Freitas. He's actually written almost, I think he's written more textbooks on the subject than Eric has. Though, truth be told, when I was in grad school, and Eric was also in grad school, we met, and I thought he was nuts. So it took him about 5 years to convince me that this nanotechnology stuff was real. A respirocyte is simply an artificial red blood cell. And the idea that Robert Freitas came up with is instead of using hemoglobin, use a pressure vessel, because pressure vessels can store many more molecules than hemoglobin can. I'll be talking about the technology, what we're developing with DARPA, and then I'll be talking about, well, what can you actually do with it? When you hear the word nanotechnology, that really doesn't mean much, because you could be talking about anything, any technology that's on the scale of 1 to 100 nanometers. What's important, I mean, size matters, but what kind of complexity you can put on that size is what really matters. What I'm interested in doing is building a closed loop of nanoscale components that can make other nanoscale components. So this is really manufacturing, nanomanufacturing. The most obvious example, which we're familiar with and a lot of people have talked about, and I'm sure you've seen a lot in your molecular biology classes, is protein engineering. Problem is we have not solved the protein folding problem, and there's no reason to believe that we will in the near future. The good thing about it is that it's low temperature. The bad thing about it is that it's not very strong. Now, one of the things, let me back up a little. One of the things that makes protein engineering so difficult is, as you can see from that center, from the center right there, is all the amino acids are connected by bonds that can rotate, and that's what makes the protein folding problem so difficult. And one way to solve that is, instead of using amino acids, you use bis amino acids, which have 2 bonds, and that way they don't rotate, and all of a sudden the whole protein folding problem goes away. This is what Christian Schaffmeister at Temple is working on. To see how you use this capability, and he's actually built quite a few bis proteins using this technique, but to actually make a productive nanosystem out of it where you can actually control what's happening is rather difficult. Just to explain, this particular diagram takes about 2 hours. So let's go on to the next one. These are all approaches to productive nanosystems. The first ones I'm talking about here are all biometric. The other really useful way of building nanostructures, or an approach to productive nanosystems, is structural DNA. And this particular technique I've illustrated here was developed by Paul Roderman, what he called DNA origami. And here we have basically a mad scientist working in his kitchen. Not really, he's actually in a lab. But he was able to come up with 50 billion smiley faces in 2 hours. A lot of concentrated happiness. And basically one person with a glorified kitchen oven. Now, to run the experiment does cost $2,000. But the real important thing about this is that we've advanced in our understanding of molecular chemistry. We've advanced our understanding what happens at the nanoscale to the point where this sort of stuff is no longer science fiction. It's systems engineering. And so basically what he did is by picking the right sequences of oligomers, he was able to make all sorts of different shapes. Not only shapes, but he was able to write letters. What we have here actually on the right is the first practical application for this technology. And what it is it's 100 trillion probes that can detect specific RNA sequences of pathogens. And then if you look really closely, the two dots over here are an index, and then depending on which bar lights up, that tells you which pathogen, with pathogens RNA you've encountered. And this is a very sensitive test. Now, in addition to the biomimetic approaches to productive nanosystems, there's also the mechanical probe-based approach, basically scanning probe microscope. Diamondoid mechanosynthesis. This is what Eric Drexler and Robert Freitas have spent a lot of time on. There's been over 100,000 hours of CPU time trying to figure out how do you take two carbon atoms, a dimer actually, and put it exactly where you want it in a particular configuration so you basically can grow diamond. Now we've grown carbon using CVD, chemical vapor deposition. We've grown diamond on thin films for quite some time now. What is different here is that you control everything to the exact atom. So this is atomically precise. This year, finally, we have funding in the UK. Professor Philip Moriarty is going to be running some experiments to prove that all these 100,000 hours of CPU time was not wasted, that they are accurate. Now, what DARPA has funded last year, they just started, is what they call tip-based nanofabrication. Basically, they want to be able to use any kind of tip-based approach to do automated parallel nanofabrication of any position, size, shape, orientation at the nanometer scale. And they want to be able to do in situ detection and repair. This is all, and they basically try to get any kind of probe-based technique that they possibly could find. One of the ones that they're funding is at Berkeley, and basically here, what they're doing is they're taking gold nanoparticles. I mean, as you know, gold melts at a very high temperature, over 1,000 degrees C. However, when it's in the form of a nanoparticle, the temperature is only about 130, 140 degrees C. So that's compatible with flexible polymers and substrates. But the problem, of course, is that you're dependent on the size of your gold nanoparticle. So they've been able to get to features about 90 nanometers, but that's not really good enough for atomic precision. At Northwestern, Chad Merkin has been working with dip pen nanolithography. The advantage here is that there are a lot of chemistries that you can put on your tip. But again, I think you're going to be limited. I mean, he's been limited to 15 nanometers so far, and that's because of the size of these needles. You can see right here, this needle. You can't get down to one particular atom at a time, which is what the group I'm associated with is with Zyvex Labs. What we want to do is we want to build 3D structures with top-down control and atomic precision. DARPA considers this the most ambitious effort, but we've been able to get funding from other places. The Texas Economic Fund, for example, is putting in a lot of money. Zyvex is putting $2 million of their own money also. The way this works is called pattern atomic layer epitaxy. And you have basically in a high vacuum, you have an STM tip that comes down and removes hydrogen atoms one at a time. And then you have a precursor gas, such as disilane, and that basically bonds to wherever you have any dangling bonds. and then you repeat as often as necessary. So this is patterned ALE. We've had some small successes so far. Joe Leiden is one of our collaborators in Illinois, and what we have on this side right here, you have to understand, here we're looking at about 15 silicon atoms wide and 6 atoms high, which is pretty impressive. Now we're getting close to atomic precision. This is a more recent photograph right here. All we've done here, we haven't done the disilane. All we've done is the patterning, and so what you're actually seeing there are the dangling bonds from the hydrogen. But it's a pretty, DARPA is very picky about their metrics, but we think we can we do a little bit more of this, and we'll be able to match the metrics they want. Now, this is as of last week. We actually did some patterned ALE. Now, it's a little tricky. For each one of these rows took about, basically what Joe Lighting did is he took a needle and he just pushed it back and forth for 10 minutes on each one to make each one of these rows. This was actually done under room temperature, under fairly high vacuum, but it's slow. On the other hand, we've got features that are 6 nanometers high. Now, one of the reasons that it doesn't look very molecularly precise is because we were having trouble with needles. Our needles just are not sharp enough, and so we're looking at some techniques for doing that. And to make them grow faster, we're going to use higher disilane pressures. Now, the big problem, of course, is throughput, and that's why DARPA is specifically saying, well, you got to use multiple probes. So multiple probe arrays, and so Zyvex is building MEMS probes. Now, the picture on the right here, this is actually done by Chad Merkin, who's also been funded by DARPA. And he's had up to 50,000 probes. We see that there's no theoretical reason why you can't have 1 million. Now, for the subject matter at hand. Robert came up with this idea back in 1998, where at that time it was pretty much science fiction. But because of the advances, it's turning into, as I said, systems engineering. He did a preliminary design study. The idea is if you have a pressure vessel, if you make it out of diamond, you could pressurize it to 100,000 atmospheres. Now, it turns out for a variety of reasons that 1,000 atmospheres is all you need. But you got to find out exactly what you're trying to do. The reason I call the Respirocyte one of the simplest nanorobots is because it doesn't have to do much. I mean, a red blood cell doesn't communicate. All it does is it absorbs oxygen and releases it at the right time, and the same with carbon dioxide. So it's simple. I mean, we're talking something that's not much more complicated than a toaster, certainly less complicated than a microwave oven. The secret about red blood cells is it's evolved some really impressive molecules, hemoglobin. Oxygen is not very soluble in water. Hemoglobin can, even though it's 68,000 daltons in terms of molecular weight, it holds 4 oxygen molecules. And what you see here is the heme molecule on the right here that actually does the work. Now, the interesting thing about hemoglobin is it has to be in red blood cells, otherwise it gets filtered out by the kidneys. And then what's special about it is that, first of all, there's a lot of them in our whole blood, but the binding is nonlinear. And because it's nonlinear, it's regulated by oxygen partial pressure, but then it changes according to pH and temperature. And in fact, it changes exactly the way you want it to. When you're in the tissues, in the capillaries, the pH has changed and becomes lowered, and so the whole graph moves to the right. And the same thing happens with temperature. So when you're exercising hard, you get hot, more oxygen gets released in the capillaries, which is exactly what you want happening. And this is from a from a computer science point of view, this is trivial. Anyone can program this. Now, there's a couple other things. If you're putting something in the body, you want it to appear non-obtrusive, so that so you don't get attacked by your own body. It turns out that the RH factor is basically not important because we're not moving ammonium across the cell membranes. RH factor we could probably fake anyone. It's just a simple protein molecule. Now, the competition is PFCs. There's been some advances recently in terms of surrounding them with lecithin. They're very efficient. Now, what they don't do is they don't trap or they don't bind the oxygen molecules. They just make it dissolve. Problem is, as you can see here, that the response is linear. And that means that you need to put almost toxic amounts of oxygen for the patient in order for the adequate oxygen to get to the capillaries. And that may be part of the reason it increases the risk of stroke in cardiac patients. And the other thing is this is a very short-term solution. It only works for a couple hours. What we would like to be able to do is have these Respirocytes in your body constantly so that you can hold your breath for 4 hours. You could have a heart attack or a stroke and not die because you have plenty of oxygen getting to your tissues. What you really need to do, what we need to do next is actually look at the subsystems that we need. And this is why I call it systems engineering. You have pressure vessels, pumps, and power. Pressure vessels, it turns out that we can build silicon today. This is crystalline thin film silicon that is quite strong. And so we can build things that can build pressure. Well, can we build the pressure vessels? We expect by the end of our DARPA project to be able to build 10 million atoms per hour. Now that's not very much, but we should be able to build pressure vessels, and it's going to be much more efficient than a red blood cell, as long as we can get all the rest of the systems taken care of. We need to know when and where we are in terms of, or each Respirocyte needs to know when and where it is. And the pressure is one indicator of where it is, so it doesn't have to have much of a memory to understand that. It's a pretty simple diagram compared to most computer programming that I've done. We already have nanosensors. Now, connecting them, integrating them is a difficult job. But we also have pH sensors, and people have actually built things, and they work quite well. And we will be able to build things like this in the end of 4 years fairly easily. Now, this particular one has been tested outdoors for 18 months. So we're pretty certain that this sort of approach will work. Now, the problem is the pumps. We've already got some people looking at aquapores and using that idea to design, here in this case, a water pump. Now, this was only, no one has actually built anything like this. This has just been tested theoretically. Same with the neon pump. But these are selective pumps. It turns out there's interesting pumps, selective pumps from Mother Nature that we might be able to get some ideas from. These are rather small. They run on ATP. Now, the thing is, well, I'll get to that later. The thing is, it's a very elegant design, but it's rather difficult to integrate it with a silicon shell. But what we would like to be able to do is basically use the hemoglobin in a more concentrated way, where you actually put it on the ends of a wheel, you run the wheel, or I should say the rotor, and then you selectively increase the partial pressure of oxygen on one side of the pressure vessel. Now, obviously, we're not going to be able to get to 1,000 atmospheres like that, so what we need to do is you have a cascaded selective rotors. Now, how do you power them? Well, again, we can go to biology and see, okay, kinesin works. It runs on ATP. It can carry cargoes. The idea is, and it's fairly efficient, 40%. What we want to be able to do with, and it uses 1 ATP per step. You can build people have already taken kinesin and attached one side of it to silicon, and then they have the microtubules move around. What if you were able to attach both of them to silicon parts, and then you can actually have a rotor? Here you have a rotor and a stator. And all you have to do is run ATP on it. If you want to increase the power, you just put more, I'm sorry. If you want to increase the power, you add more slices. If you want to increase the speed, you make more concentric motors. Now, the thing is, how do you power your motor? And fortunately, Alex has already explained all that. Here we have the famous Travis chip that actually takes your glucose, which is in the blood already, and you have it run. Now, it turns out, and I've already asked him about this, well, what about the real powerhouse? What about the Krebs cycle? And he said, well, that's really difficult. And it is, I mean, because the molecules are much bigger. There's a much, and is much more complex. One thing we might be able to do, and people have tried to do this, is they've taken mitochondria and they've attached them put them in artificial environments. So we might be able to do that. The other thing that we can do with ATP, which is kind of backwards, is you can turn it into electricity by using the ion pumps. You actually build a battery. Okay, now the thing is, okay, we talked about the oxygen. How do we get the carbon dioxide out? And this actually turns out to be fairly complicated. It turns out in our bloodstream, the carbonic anhydrase converts carbon dioxide to bicarbonate very quickly. 1 million atoms per second gets converted. This is one of the fastest reactions of all known enzymes. And basically what happens is store it. Then the question is, what do we do with the bicarbonate? We store it. And we've actually looked at the way cyanobacteria do it, and they have something called CMPA protein. It's very highly selective. The structure was only discovered 2 years ago. It's a fairly small protein, and the idea is that if you could bind this to a motor, or actually you don't, what you have to do is you have to have an environment where the CO2 is catalyzed, and then it gets transferred to a rotating wheel as bicarbonate, and then it gets moved to where you really want it to store it as you take it back to the lungs. Now, let's say that doesn't work. If we can't build selective motors, then we just build non-selective pumps, and then use selective membranes to try to keep either the oxygen or the CO2 where we want it to, and pressurize it as we want. It won't be as effective that way. That's the only problem. Now, the question: how do you control all this? I mean, I said that there's not much electronics involved, and that's true, but there are still going to be power requirements. It turns out there's a new way of doing logic, and this is called the quantum dot cellular automata. This does not use quantum physics in the traditional sense. What we have is a quantum dot where the exciton distance is smaller than is larger than the size of the dot, and so you have some interesting behavior. What happens is that by using very small electric fields, we can make the quantum dots switch state, and that's all we want, is we want it to be able to switch state. We're talking about single electrons or less to actually make this work, and people at Notre Dame especially, and I think in British Columbia, are working on this technology. It's very different from the CMOS that we're used to, but it's also 1,000 times less energy gets consumed, so we don't have to worry about heating also. And finally, the big problem, my last slide, is how do we do this? The problem is we need 3 trillion Respirocytes to treat one person, and we're only building things atomically precise at 10 million atoms per hour. So we're talking about 16 days and $120,000 to build one Respirocyte. That is just not acceptable. Now, there's a couple things we can do. So we're talking about, let's see, one person would cost $360 quadrillion. We can't, not too many people can afford that. So we got to look at, all right, what can we do about this? Nano-imprint lithography lets us lower the costs by 1 magnitude. Basically, what you do is you use nano-imprint lithography to build things, and then you use atomically precise methods to make them atomically precise, of course. And that'll get you 1 magnitude. Then the other thing you want to do is you use the scanning tunneling microscopy arrays to build larger STM arrays, and that will work for as many times as I calculated it. That will lower the cost by 2 more magnitudes. Now, we got 15 more magnitudes to go. Can we do this? It turns out one of the interesting things, even though we haven't scoped out the exact characteristics, once you've designed something that works autonomously at the atomic level, then you can increase the size. It looks like the design looks fairly straightforward. So it no longer takes 4 years to come up with the next generation. It only takes, at the beginning, it only takes 2 years, and then gets down to 16 days, and then 4 days very quickly. So I'm hopeful that we'll be able to do this. Thank you very much.
Thank you. We have time for just one question before we break for lunch.
Just a thought on the Respirocytes. In the lab of Jeff Brinker at Sandia, they've developed a construct that they call functional protocell, which is a nanoporous silica interior that he can load just about anything into, and a membrane on the surface. And he can make lots of these pretty cheap, and has gone a long way towards showing that you can independently design the interior and the exterior. So this might be an architecture for the Respirocytes that one might consider.
I like the idea of being able to do things cheaply. The thing that worries me is that it depends on self-assembly. And the problem with self-assembly is, unless you're working with DNA, is that it's very difficult to get high selectivity in terms of your reactions. So with DNA, as I showed with the smiley faces, you can get 100 nanometer resolution, 100 nanometer structures with 3 nanometer resolution. With a patterned ALE, you can get better resolution than that, but you can only do we'll be able to do 10 million an hour.
But you can coat the surface, you can put very specific proteins.
Well, the problem with proteins is they're not going to be able to hold up to 1,000 atmospheres. That's why we need to go through the crystalline silicon, or if we can, the crystalline diamond. We're running into, you have one capability here where you can have something exactly precise and get these properties, and then you have these other, you have the capability of making, billions at a time, but trying to get them to meet is going to be the hard part. And I don't see any real good shortcuts to that. On the other hand, once we can start bootstrapping, then we'll be able to get there. We'll get what we want. That's what I'm looking forward to. Thank you very much.